Hello, good day, students. Welcome again to our molecular biology lecture. I am Jason Pasao. I will be going to teach you. I'll be your lecturer for today. And currently, I am living in Poland. So, for today's lecture, we're going to highlight about the basic nucleic acid techniques, particularly the isolations and purification. And then this is the part one of our discussion. We're going to include the discussion about the genomic DNA isolation. We also need to include the plasmid DNA isolations and also your RNA isolation. For the part two of our discussion, I'm going to discuss the different methods of purification and how you're going to quantify your nucleic acids. And also, I also included on how you're going to recover your nucleic acid fragments from your agarose gel. And lastly, since it's very important for us to determine or to understand what is the principle of your gel electrophoresis, because as I mentioned, that gel electrophoresis is widely used in a molecular biology lab. So at least you have the basic foundations of understanding the gel electrophoresis. And also I'm going to introduce to you the different kinds or strategies of using the gel electrophoresis. For the learning outcomes, of course, the students should able to understand the principles of isolations of your cellular nucleic acids. And we're also going to describe the steps that is needed for isolation. So it could have in the cellular genomic level or in the plasmid DNA isolation level. The students are also expected to describe the different strategies that can be used for the separations of your RNA from your DNA. And also, additionally, we're also to discuss about the gel electrophoresis on the basic understanding of the separations using your electrophoresis. And lastly, we're going also to discuss on how you're going to recover the nucleic acid fragments from your agarose gel. So as I mentioned that I divided in different parts for our discussion today. For this part one, we're just mainly focusing on the isolation and purifications of your nucleic acids. So if you're going to isolate the cellular components, it is actually on a subsequent manipulation. Meaning to say, if you're just going to isolate your DNA in a chromosomal level, then you could isolate the nucleus. Whereas if you could isolate the DNA in the extra chromosomal level, then you can isolate plasmid, mitochondria, or your chloroplast. And also, class, you have to take note that for your determinations product of your gene expression, you could isolate an RNA or your protein. The principles of your isolations is basically same same principles with the separations. Mean to say you're going to isolate or separate one factor to the other factors. So for this one, it also applies in the isolation of your DNA or nucleic acid. So for the cellular components, it should be separated based on the size. The size usually measured in the molecular weight. Conformations, this is the characteristics or the shape of your nucleic acid. The solubility, they're going to check in a selective precipitations and also in a partitioning between the different solvents that being used. And of course, the stability, which includes the enzymatic treatment and your chemical agents. So if you're going to isolate your nucleic acid, of course, this will going to break down your cells, which is being lysed, and then they release the contents that is needed for your experiment. So you could also remove the cell membrane. Aside from that, you could also remove the cell membrane and the proteins. So you have to take note, class, that if you're going to isolate only membrane fragments, you should use a low-speed centrifugations to pellet the cell debris. Okay, and then this supernatant that we will have will contain parts of your nucleic acids and also your proteins. So if you are using different sources, because if you are performing nucleic acid isolations and purifications, you could use different sources. So as you already know that you could use plant cell, you could use animal cells or bacterial cells. So you have to take note that if you're going to use different source of your samples, you should have at least a fresh living cells, or at least the living cells. So there's also some factors that your DNA cannot be isolated because it's already damaged. One of the factor that we have here is the physical shear. So it could from the steering, you should maybe thoroughly steer your samples, or you have continuously pipette or not gently pipetting your samples. So it could also damage your DNA. And for the enzymatic activity, which is later, we're going to discuss about the enzymatic activity of each nucleic acids isolation. So for instance, we have here the nucleases. So the eventual use of your DNA will actually also dictate what methods that you're going to prepare. So if you're just going to isolate or use the DNA, which is the large fragments, then you need to require library constructions. These library constructions are your genetic sequence, okay? Or you could also use a gene isolations that is needed for a gentle preparation. Sorry for that. And if you're going to isolate only the small fragments, then you could just use the basic methods of your nucleic acids. Or like, yeah, you just follow the basic nucleic acid preparation. So the first step of your isolations is, of course, to break and open the cells and release the contents which is necessary for your experiment. So we have your different sources of your sample. It could be animal cells, bacteria and yeast cell walls, or your plant cells. So please remember that they have different characteristics that it will affect the isolation process. Okay, so for the animal cells, since they are quite fragile, then you can only break. I mean, you could. Oh, I mean, sorry. Cell membrane could be break. So we have here is very easily this prone for fast lysed. So you could use the mild detergents, and prone for osmotic shock, and you have to perform a gentle agitation. For your bacterial and yeast cells, because they are more tougher than your animal cells, then you could break down the cell membrane and your cell wall. Then, if you're going to break down the cell membrane and cell wall, then you need to have the strong detergents, which is later we're going to discuss what are those detergents that being used. And this is also prone for lysozymes and degrading. Comparing for the animal cells and bacterial and yeast cells, the plant cells are very extremely tough. Well, so you need to break down the cell membrane, the cell walls, and the lignins. So from this one, because they are very extremely tough, so you need to use a degrading enzyme such as your liquid nitrogen. For the isolations of your cellular genomic DNA, then the tissues or the cells are groundly finely in the pistils or mortals. So you need to require a liquid nitrogen that will make the tissues pre-littles and will going to facilitate the grinding. So plus you have to remember that if you're going to inactivate the DNA, so you need to use it or perform it under a low temperature. For isolations of your cellular genomic DNA, of course, you need to add the detergents. It could be a positive or negative cell wall degrading enzymes. And for the removal of your cellular debris, then you could perform a centrifugation. So you have to take note that in the supernatant, it should be in a state of a cleared lysate. And so this is the example of the cellular genomic DNA. So you have here the upper part is the supernatant, which is very cleared lysate. Usually it's transparent, and you have down below is your pellet by using these centrifugations. So for the isolations of your nucleic acid, so of course you have to perform a centrifugation because it's able to remove the membrane fragments, the organelles, as well as the associated proteins. And then also have class, you could shift to alkaline pH because most of the proteins are being denatured and aggregates. So for protein aggregations, you could use centrifugations from that to remove. For the remaining soluble one are basically your nucleic acids. So those remaining proteins could also remove through a prophase treatment, or also a solvent precipitation such as your chloroform or your aqueous interface. So this is a typical example or a graph shown to you that we have here cell lysis and the centrifugations to remove a cell debris. So as I mentioned, the principles of your isolations is to disrupt the cell wall or disrupt the cell membrane. Once it's going to disrupt, it will release the contents, which is necessary for your experiment or called as your cell extract. Then or after which you can perform a centrifugations to remove those cell debris. So we have here cell extract. You added some of the detergents or some of the reagents and then perform a centrifuge. Usually it takes 10 to 15 minutes to centrifuge this one. And then you could isolate your sample. So the top layer part contains the DNA, RNA, and protein, whereas the lower part is for cell debris. So comparing with the proteins and nucleic acid, in terms of the characteristics of abundance, the size, the acid-base characteristics, the denaturations, and your stability, there are actually differences between the proteins and the nucleic acids. So for the abundances of the proteins, there are most likely many. Dry weight of the cells so contains 50 to 60%, whereas your nucleic acids are low or just low or 10 to 20 dry weight of the cells. So both of them are actually high in molecular weight, but in terms with the characteristics with the acid base, proteins are very variable, whereas in your nucleic acids are they are acidic. So for the proteins, they are often irreversible, whereas your nucleic acids are generally reversible. For the stability status of your proteins, so they are very high, they are very stable in some conditions, and whereas your nucleic acid, they are actually in a low state or medium state of stability. Plus, if you're going to perform a protein and nucleic acid isolations, you have to take note that you should use sterile tubes or reagents or pipettes or like tips to perform this one, and you have to perform this one under laminar flow. Why? Because to avoid contaminations of your sample. Sorry. So we have here the isolations of cellular genomic DNA. So the protein is actually removed through a phenol chloroform or your isoamyl alcohol extraction. So this protein will going to precipitate at the organic or aqueous phase boundary. So your nucleic acid solutions is in the aqueous phase, whereas here in the nucleic acids, you could also precipitate this one out of the aqueous phase using your ethanol or salt precipitation because it will absorb everything. It will absorb the solvents in the salt. So these monovalent cations could actually help to neutralize the charge. So here we have here that the RNA could be removed by the RNA treatment if it's necessary. So this is the example for you to at least you have the idea what is how we're going to process with your cellular genomic DNA isolation. So we have here cell extract. Then you added a mixture or ratio of your chloroform phenol. Then you mix thoroughly about 5 minutes. Then once you mix it, you need to use centrifuge for about 10 minutes to separate the aqueous layer from the coagulated proteins and your solutions or your reagents. So at the top layer is your aqueous solutions or aqueous layer. This contains your DNA and your RNA. Okay, and then at the middle part, you can see there's a middle part because it's actually visible. It's usually it's turbid. So these are your coagulated proteins. At the lower part is your phenol or your reagent that you added previously. So this is performed through a centrifugation. There's no other way on how you're going to isolate your nucleic acids. So some of them, which is not really actually common right now. So they added glass rod to attach the DNA fibers. And then you have here the top layer is your ethanol, and then the lower level is your concentrated DNA exclusions. There's also some other way aside from that. We have through a centrifugations a very how do you call this one, I mean, kusog yung centrifugation, let's say, from that. So we have here, you could separate the reagents that you added from the precipitated DNA. No, so we have here the top layer are most like your ethanol, and then you could use the precipitated DNA for your further experimentation. So when you're going to separate RNA from DNA, of course, you need to understand the characteristics, no? Because they are very, very different. So from the RNA, so the abundance is they are most likely, there's a lot of RNA in the samples, no? You could isolate, let's say 80% of that are RNA from the samples of your nucleic acid than your DNA because they are very less abundant. So it's very difficult to isolate, more likely on the DNA. And for the molecular weight, of course, you could differentiate this one. You know, the way of this one is the RNAs are more on a lower molecular weight than your DNA, which is high molecular weight. In terms of the stability, your RNA is actually unstable, and whereas your DNA are very stable. For the selective removal, if you just want to isolate only DNA, then you could use RNase treatment, or you could have E alkaline pH. But if you are just going to isolate the RNA, then you could DNA, you could use the DNase treatment, no? So it will just remove your DNA, then you could isolate the RNA. Okay, so plus you have to take note here that your RNAs are not destroyed totally by just performing your autoclaving. So you must use a chemical treatments to prepare these tubes and reagents for your RNA solution. One of the example of this one is your RNase treatment, okay, or your alkaline pH that I mentioned here in the table. So let's talk about plasmid. I know you already have idea with the plasmid because previous discussion that we have in the prelim, we talk about more on the plasmid. So your plasmid is your extra chromosomal double-stranded DNA molecules. So it depends on the size, no? They vary on the size. It could range into 3 to 200K kilobytes, no? So this is actually plasmids are very widely used in the experiment, especially in the research, because they use or they carry like very useful genes. Example from that is that we have maybe genes for antibiotic resistance or substrate for a utilization. So since plasmid are easily replicate, they are very independently of the main chromosome. So most likely pass to daughter cells upon the cell divisions, and it could also pass between the bacterial populations. Plus also plasmid are enabled to spread the encoding characteristics. So later we're going to discuss this one on the isolation. So this is a typical picture with you to show the different how do you call shape of your plasmid. So this is the relaxed plasmid, and the other one is the super-coiled plasmid. So for your relaxed plasmid, this is actually very stable and very easily to recovered, since this is very popular when you're going to perform a cloning for your vectors, because they are already a standard characterization, a standard sequence from this relax. However, here in super coiled, though we still using this one for a cloning vectors, but we need to how do you call this one? We need to manipulate the sequences for this, because they have different like shape compared to the relax, which is actually standard already. So let's isolate for the isolations of the different forms of your DNA. You have here chromosomal DNA and your plasmid DNA. So in terms with abundance, so your chromosomal DNA are in low copy number, whereas your plasmid DNA are in high copy number. In terms with the weight, chromosomal DNA are high molecular weight, whereas your plasmid DNA are in low molecular weight. In terms with the shape or characteristics of the shape, you should remember that your chromosomal DNA are usually linear, since they are very fragmented, whereas your plasmid DNA are actually intact are very circular because most likely, as I mentioned, we have relaxed and supercoiled. No, so in terms with stability, chromosomal DNA are very prone to shear, and they are always in the nucleus attack, whereas in plasmid DNA, they are in a shape with supercoiling, and most likely they are very protective. So for the isolations of your plasmid DNA, so the most common goal here is to separate or to isolate only the plasmid and leave the chromosomal DNA with those of your cellular debris. So there are different sizes and structures of your two DNA forms exploited to separate them. No, from the plasmid DNA, they are very super coiled and small. So, mean to say they will retain their super cold formation if they are not being damaged. Whereas your chromosomal DNA, since they are large, they are associated with your protein scaffold. So these are, as I mentioned in the table, that they are subject for fragmentation and loss of a supercoiling shape. For lysis of your plasmid DNA isolation, it could be done under the alkaline conditions, no? So, but it depends now on the shape or the characteristics of your plasmid. So for a non-supercoiled DNA or your relaxed DNA, it will go into denature. I mean, sorry, the chromosome will go into denature. However, your supercoiled DNA will not denature using only the alkaline conditions. Okay, sorry from that. So if we're going to use the mixtures which is neutralized by your NaOAC or sodium acetate, the chromosomal DNA will going to precipitate. Thus, you could isolate only the cellular debris which is contained with your plasmid DNA. Okay, so we have here plasmid DNA solutions can be precipitated also using your salt or you're using salt, additionally your salt and your ethyl acetate. So here is actually cell lysis. So how about if you're going to isolate DNA isolations without using the cell lysis? So we could also do that using gentle non-ionic detergents called the Triton X100. You're going to puncture the cell wall, okay? So the cellular organelles and large chromosomal fragments are not able to release. So you're going to perform the centrifugation. So those small plasmid can exit to the surrounding buffer, then you can recover it by adding ethanol or salt precipitation. So there are different methods, no, class, as I mentioned, that there are different methods for a further purifications of your plasmid according to the need. Example, if you're going to going to determine the quality of your next phase, you're going to quantify it, you're going to minimize the protein content, you're going only to remove the bacteria and the toxins or just no RNA at all, then you could apply different purifications according to the necessity of your experiment. Now let's talk about the bacteriophage or the phage. Na? So bacteriophage is actually your nucleic acid molecules in a protein capsid. So these are contained with your DNA genome. So they are very effective at introducing genome into the host cells because they inject DNA through a cell membrane. So these phage particles are being replicated inside the effective host, and then it has many copies of your recombinant genome. So this is a typical picture for you to show what is actually a bacteriophage. I think you already have knowledge about this during your bacteriology class. So we have here the structures of your bacteriophage. We have the DNA inside. This is the head. This is the neck. This is the basic structure of the head, the neck, the sheet or the tail sheet, and then the fibers or the end plate. No, so this is the bacteriophage T2 by the sources that we have. So you can see here that we have the head, we have sort of the neck, we have here the tail, and we have here some of the fibers. You can see here. It's amazing that they could isolate the bacteriophage structures at this level. So, but also you have to remember that your phage infections will always, or let's say, it's actually very transient. I mean to say you could isolate this one in a very, very short period time, no? Very, very short lang. So that's why we have it would be divided into lytic cells or lysogenic cycle, no? For lytic cells, they are easily lyse. The cells are easily rapidly lyse. So there are many new phage particles are being released, and whereas your lysogenic cycle, these are the phage DNA being integrated into host DNA, and then there's also some of the retained for many cell divisions, and these lyses were being induced at the later stage, and then it increases the numbers of the cells being infected. Therefore, this phage is particles is being produced. So this is the um the principles or the overview of your reproductive cycle of your bacteriophage. We have here the Wilhelm phage stage, so which is your bacteriophage being attached to the cells, then they're being injected some of the particles, then it will replicate here under the cells, and then it will release to the our body or the disease system to destroy the cells. Whereas your lysogenic reproductive cycle for your bacteriophage, you have here the temperate cells, the prophage, no? So the injected, the released, then we have here from the lactic cells. However, here in the lysogenic, the replicate the phage. So that's why it is prone for a it increases the number of the cells infected. So for the phage DNA isolations, we have here pure phage particles that is easily isolated by the density gradient of your centrifugations. So we have here phage particles that are suspended in the appropriate medium. So you could use proteinase K if you want to isolate phage protein coats are digested using your proteinase K, then it could actually help to facilitate the protein removal. And then also you could use a phenol ratio of your phenol chloroform instructions that could remove the cold protein, and then you could have isolate your only the phage DNA, which is you adding your ethyl acetate. Again, class, let's just review about this one, that you have going to isolate only the phage isolations now. So you could add first a proteinase K, and then you could remove some of the protein. Then you have to add phenol chloroform to remove the cold protein, and then you have to add the ethyl acetate to precipitate your phage DNA. Then you could use this one for your experiment. So I want to show you some video about the supplementary about the phage cycles that I mentioned here. But since we don't have enough time, so maybe I just have to give you the references and you could watch it at your own pace. Huh? So this is the different kits for the DNA, isolations. So we have here quadrant, which is widely used in the laboratory. We have also here filter. We have also high-speed plasmid kit, Indo-free plasmid kit, and the quadrant large construct. So I am actually more on using this quadrant plasmid kit because compared to other, these are more cheaper, and this you could just usually replace it easily. Yeah, you could just easily replace it, and it's common in the laboratory as well. But I tried some of the Indo-free plasmid kit, but it's tricky and not like just a lot of things like to perform. But this quadrant plasmid kit is like easily, dali lang kayo siya, very convenient for science and molecular biologists who are performing numbers of samples, to examine. So we have here the quadrant plasmid kit. We have pellet bacteria, then you have you have alkaline lysate, and then you have clear lysate. By using your centrifugations, it will bind the DNA. Then you have to wash it using your buffer. Then you have to add elute or elutions, solutions. Then you could add isopropanol, and then you could collect the DNA by your centrifugations, So you use the ultra pure plasmid DNA. So however here, let's compare with the other one, because some of the other kits, commercialized kits that was in the laboratory use different, example, they use clear lysate by filtration, which is actually good because it could more like isolate, or you could collect more like the abundance or the samples that you could collect, the number of samples that you collect is better way than the quadrant plasmid kits, because they used a filter, cold house vacuum, same with the plasmid kits, So and then however here in the Indo-free plasmid kits, we add some of the endotoxin or removal buffer, no? And then we need to incubate this one for 30 minutes. So it takes a lot of time to perform endo-free, but I think compared to Quagen, endo-free is more like, let's say, more accurate, or you could isolate more of the protein or nucleic acid compared to Quagen kit, no? So, but this is actually not common in the laboratory because we don't know it's expensive and it takes a lot of time. But if ever, if you're just going to isolate very important like sample, then you could use in the free plasmid kits. No? So for the part two of our discussion, we're going to discuss about the purifications and quantifications.